Bismillahirrahmanirrahim. In last lecture, we just discussed about multiple sequence alignment and different dynamic programming alignment and pairwise sequence alignment. So now we will try to run Clustal Omega and just have a look. So first of all, we just need some sequences. And here I have UniProt, and if I will just write here any protein name, so we can just take the sequence of protein from here. Like this is the protein sequence, and I will just take the faster format. As you guys knows very well that Clustal Omega just take the faster sequence. So I just copy three different sequences, and make a file. I just copy the sequences, and I will open Clustal Omega. If you will write just simple Clustal Omega on Google, you will have this first link, that is www.ebi.ac.ck, and you will have this Clustal Omega software tool. So multiple sequence alignment. For multiple sequence alignment, we have three steps. In the first step, we have to enter our protein sequences. So you can also use this for DNA and RNA sequences. However, we just prepare protein sequences. So we are just going to paste three different protein sequences here in FASTA format. We can also upload the file by this option, upload the file and use this. After that, we have the second step, that is set for your parameters. Usually we use default parameters. However, if your nature of experiment is different, you can change the parameters. And on the third side, you just have to submit the sequence. So we have only three sequences, so maybe the result will generate very soon. However, if you have more than 10, 20, 30 sequences and the length of sequences is 500, 600 amino acids, then it will take much time. So better to notify your email. So if you just shut down your computer or you are busy somewhere else, the result will be, the software will mail you the result on your email address. So I just submit it. And your job is currently running. Please be patient. So we will show good patience here. After that, we will have the result. Yes, now I have the result. Because it was only three sequences, the result came very early. However, when it will be more sequences and the length is much more, then maybe there is some problem. So we can see that here we have three sequences, 1, 2, 3 from first amino acid to 60, 60, and then. So you can see that here is the conserved part here. These static just show the conserved part, that this is the conserved part of all three protein sequences we are going to use. You can also analyze motif from this domain from this, or you can also try to hunt a family of unknown protein. So we already discussed all this in our previous lecture. So static mean that these are the conserved path. Then there is a gap sometimes that is not have any similarity. These two columns also, the column also show the similarity in the amino acid that has good physiochemical properties. So you can study the result. So here we have the result summary also from the second option. You can study that which guide tree it generate, which type of phylogenetic tree, which tool. This is the guide tree. As we already discussed that for analysis, we just need a guide tree. In our previous lecture, we discussed it in detail. So here you can see the guide tree just guide us that which two proteins first align, and then after that other one. Then we have a phylogenetic tree generated with these sequences results reviewer. After that we have yes, you can download the alignment sequence from here. So you just use it as presented here. You can also have a detailed look with the colors. The colors just explain every amino acid has a specific color, and the color just to make the results more beautiful or prominent. So it is very easy if you are good in literature and if you know which sequences you are going to submit and what will be the result and how to analyze the result. So I hope this is very easy, and I hope you guys understand it very well. Just open Clustal Omega, paste the sequences and analyze. Thank you so much.